Hello again, everyone. Today we're going to be talking about complex lipids. Some of this is a little bit of a review, talking a little bit about phospholipids and cholesterol. We're going to dive a little bit deeper into some of those, talk a little bit about the biosynthesis of cholesterol and derivatives of cholesterol, including steroids, bile acids. And then I'm going to end the lecture talking about lipid transport. The complex lipids we're going to focus on are glycerol lipids, sphingolipids, and cholesterol. These are typically found embedded in two locations, either in biological membranes, shown by this diagram here, or in circulating lipoproteins, which I've introduced in the past and we'll talk a little bit more about today. As a review, glycerol phospholipids are polar lipids derived from phosphatidic acid, also known as 1,2-diacylglycerol 3-phosphate. So they have a phosphate group that's esterified to a glycerol, and then there are two fatty acids that are attached to that. Attached to the phosphate, you have your polar head group. It can be things like choline, serine, ethanolamine, or signaling molecules like inositol. So again, just a review here. This is in contrast, a little bit in contrast to sphingolipids, where the backbone is not glycerol. The backbone is a long-chain amino alcohol called sphingosine, shown by this dark gray box here. And there's a fatty acid attached to the amide linkage here, attached to the carbonyl that's the amide linkage to this sphingosine head group. And then, or the sphingosine backbone, excuse me. And then you can have various head groups as shown here. If you have just a hydrogen there, it's ceramide. If you have phosphocholine, it's called sphingomyelin. And you can see all these here. These sphingolipids are found largely in the outer face of the plasma membrane. They can have carbohydrate structures that attach to them that face into the exterior of the environment, and this is where we get our red blood cells for recognition of blood type, as I've discussed in the past. Interestingly, the name came from, sphingolipids came from the fact that they were sphinx-like just because they were unknown for a long time, so that's why they were originally called sphingolipids. As I mentioned, the difference is that amide linkage for the difference between phospholipids and sphingolipids. Therefore, glycerol phospholipids can be hydrolyzed by a strong base. By adding something strong base like sodium hydroxide, you get release of glycerol and the free fatty acids. Where if you have this amide linkage, you don't get that. Hydrolysis. When we talk about glycerol phospholipid synthesis, I've touched a little bit on this, but it occurs in all animal cells except for red blood cells. The glycerol phospholipid synthesis, the precursors are phosphatidic acid and 1,2-diacylglycerol. These are common intermediates in the synthesis of both triglycerides and phospholipids. Glycerol 3-phosphate is the primary starting material for the synthesis of phosphatidic acid, whether you're starting in liver or in adipose tissue. The route to which you get to that point is a little bit different between the two tissues. But after you get glycerol 3-phosphate, then you can make phosphatidic acid, diacylglycerol, and so on, triacylglycerol, and so on. But you can make phospholipids from phosphatidic acid, as we've talked about in other lectures. To get a little bit deeper into phosphatidic acid synthesis, the main steps are shown here. You can have two different versions of the synthesis. So you can do it through glycerol 3-phosphate acylation, and that's shown by this pathway here. So what happens here is you first have glycerol 3-phosphate, and you get transfer of one long chain fatty acid from fatty acyl CoA, shown here, to form lysophosphatidic acid, shown here, and then you get an unsaturated fatty acid added to the second carbon to form phosphatidic acid. You can also get to this route starting from dihydroxyacetone phosphate by adding an acyl group to the 1-hydroxyl position, and then you get a reduction and then an acylation to end up again with phosphatidic acid. And then from here, the final step, you can form diacylglycerol from phosphatidic acid by a phosphatase. Then off of that, you can form triacylglycerols, other kinds of phospholipids as well. Phosphatidylcholine is one example of another phospholipid. It's also known as lecithin. In this case, the choline head group is converted to phosphocholine, shown here through the hydrolysis of ATP. You get a phosphate from ATP to form phosphocholine. And then phosphocholine is activated to CDP choline by this pyrophosphorylase. Then in the third step, the pyrophosphate bond is cleaved and phosphatidylcholine is transferred to diacylglycerol to form the phosphatidylcholine. So really the important things here to understand is you get addition of a phosphate group from ATP and then this activation by CTP. You can also get conversion of phosphocholine to phosphoserine by what's shown here by this primary pathway, where you get phosphatidylcholine to react with free serine to form phosphatidylserine, and then you release choline as the free base. This choline can be activated to CDP, which was shown on the previous slide, and other phospholipids can be synthesized in this pathway as well. The secondary pathway, you can also get phosphatidylcholine through multiple steps where you get decarboxylation of phosphatidylserine, shown here, to form phosphatidylethanolamine, and then you get essentially 3 methyl groups attached through S-adenosylmethionine. You can also have alcohol-containing phospholipids, such as phosphatidylglycerol and phosphatidylinositol. In this case, what happens is you get activation by cytosine nucleotides. So again, you see this step where you get activation through CTP, but in this case, the phosphatidic acid is activated rather than the head group. So then you get CDP diacylglycerol. And then in the second step, the phosphatidic acid group is transferred to free glycerol or inositol if you're making phosphatidylinositol to form phosphatidylglycerol, shown here, or you can swap this for phosphatidylinositol on the analogous reaction. So again, you get activation activation of the phosphatidic acid by CDP, and then the phosphatidic group is transferred to free glycerol. Cardiolipin is a unique phospholipid. It's essentially by attaching a second phosphatidic acid to phosphatidylglycerol to form 1,3-diphosphatidylglycerol, or what's more commonly known as cardiolipin, shown here. So it has a phosphatidic acid and a phosphatidylglycerol moieties to it. It's found almost exclusively in the inner mitochondrial membrane. It's called cardiolipin because it represents about 20% of the phospholipids in the heart mitochondria. This is a very important lipid because it's required for efficient activities of the electron transport complexes and the ATP to DP translocase. That's how phospholipids are made at a very high level, simplistic level, just to give you a background on that. Phospholipids can also be turned over by phospholipases. There are different phospholipases, and they are named differently depending on which bonds they interact with. So for example, a phospholipase A2, as shown here, cleaves this fatty acid here. The second fatty acid, phospholipase A2 and phospholipase C, these are active during inflammatory response and in signal transduction. Phospholipase B, which actually isn't shown here, but what it does is removes the second fatty acid. It's a lysophospholipase that removes the second acyl group after the action of phospholipase A1 or A2. So after you get the first one off, phospholipase B will take the second one off. These lysophospholipids may be degraded into phosphatidic acid or recycled by scavenger pathways. Just want to mention glycolipids as well. These, you'll see the terminology glycolipids sometimes. These are sphingolipids with covalently bound sugars. So they're known as glycolipids or glycosphingolipids. So these have a polar head group comprising the sugars, which is attached to a ceramide by a glycosidic bond at the terminal hydroxyl group of sphingosine. So I introduced these when I talked about sphingosines a little bit, but there's a subset of these that are called glycolipids. As I mentioned in the past, these are important for our blood group typing. So the blood groups are determined in part by the types of sugars that are located on the head groups of these glycolipids. So if you have, if you're shown our generic red blood cells with the glycolipids on the membrane here, if you have N-acetyl-galactosamine group shown by this purple hexagon here, then you are the A blood type, or you have the A antigen. If you have the B blood type, you have galactose. If you have both of them, then obviously you're the AB type, so you have the purple and the tan here. If you have neither one, you are the O antigen, or the O blood type. It can get a little bit more complex than that, but at a high level, that's the gist of the blood types and how they are related to glycosphingolipids. That's a little bit of a review, and there's a little bit of new information there, possibly from some of your old lectures, but just wanted to reorient you on complex lipids, in particular phospholipids and sphingolipids. The other lipids we've talked about in the past and I'm going to dive a little bit deeper into is cholesterol and compounds that are related to cholesterol, such as steroids and bile acids. Here's a picture of cholesterol itself. It's a six-membered ring, and it has this fatty tail. Cholesterol is an essential component of mammalian cell membranes, and again, it's precursors to bile acids, steroids, and vitamin D. The highest concentrations in plasma membranes is in plasma membranes. It's up to about 25% of the lipid content, and it's virtually absent from the inner mitochondrial membranes. And as we've talked about in the past, this can transfer in and out of membranes because it's so hydrophobic. We get cholesterol obviously from our diet, and it's transported to the liver and tissues in chylomicrons, and we'll talk about that here in a minute. But we also make cholesterol. So we make about 1 gram of cholesterol per day in our liver. In plasma, cholesterol is incorporated into lipoproteins as cholesterol esters to form cholesterol lecithin by cholesterol lecithin acyltransferase. So you get esters of cholesterol by this enzyme here. In tissues, you'll see cholesterol stored as esters that are formed by acyl-CoA cholesterol acyltransferase. And we make about 1 gram of cholesterol per day. Luckily, we excrete about 1 gram per day of cholesterol is eliminated in the feces. About half of this is excreted as bile acids, and we'll talk about what those are. And the remainder as neutral sterols produced by bacterial reduction of the cholesterol molecule. And we'll talk briefly about that as well. Cholesterol is highly regulated in our body. It's the reduction in activity and expression of HMG-CoA reductase decreases cholesterol synthesis. HMG-CoA reductase is the first committed step in the pathway to making cholesterol. So if we either reduce its activity or reduce its expression, you get decrease in cholesterol synthesis. It's also regulated through LDL receptors. So if you get downregulation of LDL receptors, it limits further cellular entry of cholesterol. You can get increase in cholesterol and phospholipid efflux from cells to apoproteins as part of cholesterol regulation as well. If you get increase in the rate of conversion of bile acids, so you get more excreted, this is a way cholesterol is regulated as well. Let's talk briefly about cholesterol biosynthesis. Don't expect you guys to memorize the pathway, but there are some important notes here to keep in mind. So this pathway begins with 3 molecules of acetyl-CoA that are converted into a 6-carbon malavinate. So shown here. So you're ending up with 3 molecules of a 2-carbon unit to form a 6-carbon unit. This happens through 2 condensation steps using acetoacetyl-CoA thiolase and HMG-CoA synthase. And then in the final 3rd step, which is the most important step, is you get irreversible formation of malavinate by HMG-CoA reductase to form malavinate here. So this is regulated by insulin, glucagon, and cortisol. There are drugs that target this, as we'll talk about here in a minute. So if you take anything home from this slide, this is the most important step here. So the drugs that inhibit HMG-CoA reductase are known as statins. Mevastatin, for example, this was isolated from a bacterium, Penicillium, and lovastatin is another one that was isolated from Aspergillus. These inhibitors decrease the intracellular. The mechanism of these statins works by decreasing intracellular cholesterol concentration and increasing the expression of LDL receptors. This LDL clearance increases. LDL clearance increases and circulating LDL cholesterol decreases when this happens. Interestingly, these drugs are taken at night to ensure maximal effect. This is because your HMG-CoA reductase in your liver activity peaks about 6 hours after dark and goes down about 6 hours after exposure to the light. The inhibition of these inhibitors of HMG-CoA reductase are typically competitive inhibitors of HMG-CoA reductase. They mimic the intermediate of the reaction, and they prevent binding of the HMG-CoA substrate to the enzyme. So the steps beyond that get pretty complicated, but I just wanted to give you an overview of some of the highlights here. So after forming malavinate, you get these isoprene groups, adding more and more isoprene groups to form farnesyl, and squalene is a big intermediate here. Some of these middle steps can lead to other things in our bodies, things like prenylation of proteins, ubiquinone, which is important in oxidative phosphorylation, as we talked about, the electron transport chain. So this pathway is important for not only cholesterol but other molecules in our body as well. But ultimately what happens is from the squalene intermediate, you get linosterol and ultimately cholesterol. This is, as I mentioned, a fairly complicated pathway, but essentially what happens is you get 3 molecules of malabonate decarboxylated into 5 carbon atom isoprene units, and these condense together to form pyrophosphate, as I showed on the last slide. Then you get further condensation to get to a 15 carbon unit of farnesyl pyrophosphate molecule. Then squalene synthase is an important enzyme. It condenses these two 15 carbon units to the 13 carbon unit squalene. And then squalene cyclizes to linosterol, which is also 30 carbons. It's just a cyclic form of squalene. And then you get conversion of linosterol to cholesterol by 3 decarboxylations, an isomerization, and a reduction. And then NADPH is consumed in 4 of these reactions. Here's a picture of some of these. Again, I don't expect you to memorize these pathways, but I do think it's helpful, at least for me, to visualize it. But you get buildup of squalene cyclizing to linosterol, and then ultimately to cholesterol. As I mentioned, cholesterol is important for making bile acids. So what are bile acids? The liver removes cholesterol as a free form or as these bile acids. Here's a picture of some of these bile acids. You can see that they're very similar in structure to cholesterol. They're just more hydrophilic. You can see more decorations here. There are 4 primary bile acids shown here, which are made in the liver. The terminal 3 carbon atoms of the cholesterol side chain are removed, and then you get this saturated steroid nucleus, so you don't have the double bonds anymore. And then you get these hydroxyl group decorations, as I mentioned. The bile acids are found in the bile, which is the intestine fluid that aids in digestion. So what these things do is they assist in digestion of dietary fat. Essentially what they are is detergents. So they have the polar carboxyl and hydroxyl groups as showed on the last slide, and then they have the hydrophobic components that the hydrophobic rings. So you get emulsification of ingested lipids through the use of these bile acids, and that helps solubilization and enzymatic digestion and absorption of dietary fat. About 30 grams of bile acids can pass into the intestine each day. The total bile acid pool is only about 3 grams, and you get about 2% lost in the feces. What happens is the majority of these are deconjugated and resorbed into enterohepatic circulation about 5 to 10 times per day. But the important thing is really up here of what the function of the bile acids are. As I mentioned, there's primary bile acids, but then you might have seen on the previous slide there are also secondary bile acids. So the primary bile acids are made in the liver, and then they're transported to the intestine. And these secondary bile acids are formed in the intestine through the action of anaerobic bacteria, so they can enzymatically act on the primary bile acids to form secondary bile acids. The rate limiting step in the synthesis of bile acids is the first step in which cholesterol goes to 7-alpha cholesterol by 7-alpha hydroxylase enzyme, which is also known as CYP7A1. The expression of this enzyme is controlled by the amount of bile acids returning to the liver and dietary bile acids. It's also controlled by gastrointestinal hormones, such as those listed on the slide here. That's bile acids. Cholesterol is also important for making steroid hormones, so cholesterol is a precursor to all these, and some of these are shown here. There's three main groups: corticosteroids, androgens, and estrogens. So essentially, the presence and the positions of various double bonds and the orientations of the hydroxyl groups or other functional groups are characteristics of individual hormones. So you can see these are just slightly different from each other. Get my pen working here. Just by the decorations that are on the ring here. The biosynthesis of these steroids occurs only in three organs in our body, and the corticosteroids are made in the adrenal cortex, androgens are made in testes in men, and estrogens are made in ovaries in women. So you can see some of the pathways here. The intermediates aren't terribly important, but all three organs are capable of secreting small amounts of the steroids belonging to the other groups, but they mainly secrete and make the ones that they're shown here. Steroids are excreted via the kidney and the urine. There are two main steps involved here. First, the biological potency of steroids removed by a series of reduction. So essentially you reduce the molecule so they're not as potent anymore. And then secondarily, the steroid structure is conjugated to polar molecules to make it more water soluble so it can be secreted in the urine. That's cholesterol. In the last 10 or so slides, I'm going to talk a little bit about lipid transport, again at a pretty high level. As I mentioned before, lipoproteins are composed of cholesterol, phospholipids, and triglycerides. There's also apoproteins in here. So these things are carried throughout our body, and our cholesterol levels are a measure of these things. So they are obviously very important clinically. So I'm going to talk through a little bit about the processes of how these are classified and how they are transported throughout the body. First, the classification. There are a couple terms you'll see here. All of these are lipoproteins, but they're just different classes of lipoproteins. So when we say chylomicrons, the main component of these are triacylglycerides. They carry cholesterol and triacylglycerols from the liver to adipose tissues, and they also take in cholesterol from the diet. So they are low in density, and they are since they mainly contain triacylglycerols. There's also things called very low density lipoproteins. The main component again is triacylglycerides. These are just a repacking of chylomicrons when they are repacked in the liver. So they are changed from chylomicrons to VLDLs when they are repacked in the liver. There's also what you may see as intermediates or IDLs. These are triacylglycerols and cholesterol remnants. LDLs, or low density lipoprotein, here the main component is cholesterol, and they are important for carrying lipids around the body. The HDLs are what you think of when you think of the good lipoproteins. These are the high density lipoproteins because the main component is protein. They carry phospholipids, cholesterol, triacylglycerol, and the other components of the HDLs back to the liver where they are transferred into bile acids and secreted. So these are the good ones because essentially you're getting rid of a lot of the cholesterol and triacylglycerides. Lipoprotein receptors are important when we talk about lipoprotein transport. The main one being the LDL or ApoB receptor. It specifically mediates cellular uptake of intact LDL particles. There are also things called scavenger receptors, which can bind many different molecules, allowing internalization of chemically modified, such as oxidized LDLs. So these will recognize a wider range of lipoproteins. The expression of these guys are regulated by intracellular cholesterol concentration, where for the LDL receptor, whereas the scavenger receptor remains unregulated. Let's talk a little bit about lipoprotein metabolism. Here's the high-level steps. So what happens here is you get assembly of lipoprotein particles. So chylomicrons are assembled in the intestine after you eat a meal, and in the liver if you're fasting. The transport of fatty acids from lipoproteins to cells then occurs, and it transforms chylomicrons and VLDLs into remnant particles. You then get the binding of the remnant particles to receptors and their cellular uptake. You get transformation of some of the remnants into LDLs, their subsequent binding to VE receptors and uptake, and then the reverse, where you get cholesterol transport or removal of cholesterol from the cells by the HDL particles. So we talk about your cholesterol levels and your triglycerides. The marker of the activity of these, the fuel transport pathway, which we'll talk about, is the measurement of plasma triglycerides. So let me explain a little bit more what I mean there. Here's a picture of essentially what the last slide just said. But you get, I'll talk through each of these, but this diagram will get smaller on the next slides. But the dark blue lines being what happens essentially during the fed state, and then the lighter blue lines during the fasting state, and then we also have something called the overflow pathway, which I'll talk about. So all these things are going on all the time in our body, but during the fed state, more of this is occurring, and during the fasting state, more of this is occurring. So let's talk through that. So during the fuel transport pathway during the fed state, what happens is chylomicrons are formed in the intestine. They transport triacylglycerols to peripheral cells, shown here. Then this enzyme called lipoprotein lipase hydrolyzes triglycerides to fatty acids. Then the chylomicron remnants are metabolized in the liver by binding through the ApoE, by binding through the ApoE or to the LDL receptor, I should say, or this LDL receptor related protein, which is shown here, LRP. So that's the fed state of the fuel transport pathway. Then during the fasting state, what happens is you get VLDLs transport fuel from the liver to the peripheral tissues, and then the VLDL remnants return to the liver and are metabolized, shown here. So then approximately about 65% of the VLDLs are taken up by the liver, and the remaining ones are hydrolyzed by hepatic triglyceride lipase, shown here, yielding LDL particles. So you get LDL particles formed at the outset of this. This is where the overflow pathway comes in. So we need further LDL metabolism occurs through this overflow pathway. So as I mentioned, these LDLs are generated from remnants in the fuel transport pathway. They can be rich in cholesterol. They are rich in cholesterol. So what happens is they are taken up by the ApoBE receptor in peripheral cells. There's a YouTube video I found a while back that explains this and kind of shows it more live in action. If you're interested in watching that, it might help think through that a little bit. The last slide here is the study guide for today. So just to reiterate some of the highlights of today's lectures that are most important for the exam, because I realize that's really what you guys are caring about here. But the take-home messages, I want you guys to understand the major phospholipids and the general synthesis of them. So I mentioned 3 or 4 of the major phospholipids. So again, I don't expect you to draw structures or anything like that, but I do want you to understand a little bit the differences between them, or minimally by name. If I say phosphatidylcholine, you should understand that that is a phospholipid. I talked through a fair amount of synthesis at the beginning of the slides. I wouldn't worry too much about that, but some of the major steps, things I highlighted in red, may come up in the exam. The difference between phospholipids and sphingolipids, I harped on a little bit. That could come up as well. The role of sphingolipids in blood types, that is an important point I've made. The main building blocks of cholesterol, HMG-CoA reductase came up a lot. That could show up on the exam. How cholesterol is regulated, how it's inhibited. Again, HMG-CoA reductase being a target there. And then I talked about bile acids. So the main things there are what the function of bile acids are, where you find the primary and the secondary, and what the differences are there. Not necessarily the structural differences, but how they are made. One's made in the liver by our own enzymes, the other one's made in the intestine by anaerobic bacteria. The three main steroids I discussed and how they are secreted, that could come up. A slide or two on that. The main lipoprotein classes and how they're different, particularly how HDLs are different from the other ones. The lipoprotein receptors, I talked a little bit about that, and then I left you guys with the fuel transport pathway. So the main difference between the fed state and the fasting state for the fuel transport pathway, and then why is there a need for the overflow pathway? Essentially because of the further metabolism is needed for LDLs. That's all I have today, so I appreciate your attention, and I will see you next time.